Welcome to our lab. Here in Munich, we are tackling the next big milestone in biomedical manufacturing. The idea of bioprinting organs has been intriguing researchers and medical doctors around the world for a while now, but the technology has yet failed to progress from basic research to application. This is where we desire to make a contribution. First up, the device. Like every piece of equipment in the lab, current bioprinters are pretty expensive. But they don't have to be. We designed an open-source DIY bioprinting platform based on the Ultimaker 2 Plus.
For our hardware expansion pack, all you need is the printer, filament and some mechanical parts. The main components are the dish retainer that fixes the reservoir, the print head that holds the needle, and the syringe pump that delivers a certain volume flow of our biotINK and communicates with the printer. How that works on a molecular level becomes clear when magnified.
Regular cells in suspension will diffuse and at times make contact but not readily adhere to one another. In order to illuminate how we enable formation of a stable cell mesh, let us pause here and first take a closer look at our printable cells. Our cells are modified to display a modular transmembrane protein on their surface, which forms one part of a two-component system. We designed 3 such proteins, all of which are capable of initiating cell-to-cell cross-linking based on the extraordinarily strong biotin-avidin interaction. To render our membrane proteins modular, they share the same intracellular and transmembrane domain. The former is mRuby3, a red fluorescent protein for visualization. The latter is taken from the epithelial growth factor receptor, or EGFR for short. The 3 deviate only in terms of their extracellular domain, which confers functionality in our bioprinting system, and thus in part differ in their working principle. For the EMA membrane protein, this domain is enhanced monoavidin. For the SCA membrane protein, it is single-chain avidin. Both are biotin-binding proteins, with EMA being mono and SCA tetravalent. The biotin acceptor peptide, or BAP, membrane protein's extracellular domain is nanologue luciferase with the BAP, also known as AVI-tag, fused to its N-terminus. This tag is biotinylated in vivo by the co-expressed bacterial biotin ligase BIR-A, making this protein bear biotin at the cell surface. Now, these membrane proteins alone are not sufficient to induce polymerization so that the cell-containing bioINK stays liquid. Formation of the cell mesh occurs only during printing. Let us first look at polymerization using the biotin-binding EMA membrane protein. When printing the cells into the reservoir, the second component of our two-component glue comes into play. Pas-lysine, a 200 residue polypeptide composed mostly of proline, alanine and serine, with 1 lysine in every 20 amino acid repeat. These lysines are chemically biotinylated. Once a molecule of pas-lysine approaches the cell's surface, the EMA protein can capture it via one of its biotin moieties. The off rate of this association is so small as to be negligible. Since pas-lysine carries multiple biotin residues, another cell that approaches the first can capture a further biotin, creating an exceptionally stable, though non-covalent, link between them. SCA being a biotin-binding protein as well, the working principle of the SCA protein is much the same. However, with the BAP protein, we are dealing with the reverse situation. The second component needed for association is the biotin-binding protein streptavidin, which binds to the BAP receptor's biotin residue. Since streptavidin is tetravalent, it can now still bind a second cell's receptor. Alternatively, biotinylated pas-lysine is added to the bioINK so that cells are connected via a bridge of at least one streptavidin pas-lysine streptavidin unit. Now that we understand the principle of our bioprinting approach on a molecular level, let us have a look at how this works on a cellular level. When we add biotinylated pas-lysine to cells bearing the EMA membrane protein, The cells stably adhere to each other once they make contact, so that eventually they aggregate into a mesh.